In this session then we're going to have a look at the process of crossing over in detail that takes place in prophase 1 and sort of continues through into metaphase 1 of meiosis. Remember, this is the another mechanism by which genetic variation is generated within the daughter cells, the gametes of the organism, along with independent segregation. So if I use the same example that we've been running through so far, we've got this organism that's got diploid number of 4 chromosomes, and we're using different colours to represent the maternal and the paternal chromosomes. So we've been using orange and brown for one of them, and then a lighter green and a darker green for the other homologous pair of chromosomes. So they're present within the cell. We know that during interphase, those chromosomes are going to carry out semi-conservative replication, and therefore our chromosomes will now look like this. In meiosis, in prophase 1 and then into metaphase 1, each of those homologous pairs of chromosomes pair up, and each of the homologous pairs of chromosomes are then referred to as a bivalent. So if we have a look at, like I said, in a bit of detail then in terms of what's actually happening in prophase 1, where the homologous pairs of chromosomes pair up, the inner chromatids from each chromosome that are facing one another get very close, and therefore they can and they do actually come into contact with one another. And this all takes place then in prophase 1. Now I'm going to exaggerate some of this just to help with illustrating what's occurring. So from our first bivalent, the inner chromatids have physically come into contact with one another, and I'm going to draw exactly the same for the second bivalent. And so what we can see then here is that this point where the maternal and paternal DNA from the chromatids facing one another, singular we'd call that a chiasma, but in this case then we've got 2 chiasmata that have occurred. Now I've shown them in the exact same position, and I've only shown one on each of the bivalents, but the reality is that they can occur anywhere. There can be multiple ones, there can be hundreds of thousands of chiasmata, and theoretically there might be none. It's a completely random process. So that's the chiasmata forming, so the physical touching of the maternal and paternal DNA. And then to illustrate crossing over taking place, remember, crossing over is where the maternal and paternal DNA breaks and then is exchanged between the homologous pairs of chromosomes. So we'd have something like so, and with our other bivalent, we would have something like this. So what we can see here then, this is now crossing over has been completed. And there and there. What we formed then here is what's referred to as recombinant DNA, or it's a recombinant chromosome. And so recombinant refers to the fact that you've got DNA from two different sources within the same chromosome, and this is the second source of genetic variation on top of independent segregation. As we've seen previously, independent segregation is responsible for giving the gametes different possible combinations of both the maternal and paternal chromosomes, and then this is amplified massively by the fact that on each maternal and paternal chromosome, due to crossing over, we can have individual genes or several genes in different loci that end up being mixed up as well.